So thank you, Marla, and thank you. Make sure you thank Casey for us, please. The introduction to that video is meant because the next talk is a long old friend of mine and colleague, Varda Rotter, who's been in the same sense that Dr. Strong is one of the pioneers in the field of Li-Fraumeni syndrome, so is Dr. Rotter one of the pioneers right from the beginning in the field of P53, biology, genetics, and understanding. And so we thought it fitting that she give the first lecture of the afternoon, which is also named and as the Tanner Project keynote lecture, and that's where the link comes. So Varda comes from Israel, not quite as far as Malaysia, but about half the way, I suppose. And at the Weizmann Institute there, a fundamental basic science institute that is leaning towards translation of basic science to clinical work, and she will tell us about that.
Thank you very much. Thank you for inviting me and letting me have a view close to you people on what I'm working so many years, but it never really struck my mind that it has to do really with human being. I'm, as David said, a basic researcher. My focus is always to solve problem and use mice as a tool to fight, as a tool to manage. But a few years ago, I was invited by David to Toronto for the Li-Fraumeni meeting, and it struck me that behind all these experiments, wonderful experiments we are doing in the lab, there is really an important cause, and that is to help people who have cancer. And in particular, I think there is great hope and thought for people who are suffering Li-Fraumeni. And I was listening to you yesterday, and obviously, you cannot just go over and say, I'll forget about it. Obviously, I will carry it, I think, for many years. And this is why last night I decided to change my talk. I had a very sophisticated talk of how P53 is working, etc., etc. But then I thought maybe I will just bring you a few simple slides to tell how P53 started. And I devoted, obviously, to the wonderful Katie that you just saw. And there is another nice young woman here in the audience, Lexis, which that talk is really meant to explain to her what is P53. So, whoever knows and whoever is knowledgeable, please excuse me. I'm just talking from my heart to the heart who wants to listen. So, I'm too excited to make it work. It's not working? I don't know. Oh, no, how do you pass the slides? They're not working? Yeah, it doesn't work. The green arrow is not working? Okay. So when we are talking cancer, the most important word that comes to mind is genome fidelity. Our normal cells are very loyal to the DNA that they carry. They cannot suffer mistakes, and they cannot suffer especially mistakes which have to do with growth control. So you are a normal cell, you are dividing, you are expressing the content of you know how to do, but you have to know to read the language properly. You cannot afford mistake in the genomic language. So cancer can be regarded as the disease of our genome. Sometimes the genome gets confused, sometimes the genome doesn't read correct the letters, and this is where you develop in many times cancer. This is one way. And the other way is really that cancer, these mistakes can be transmitted by the generations, like the color of your eyes, the predisposition to develop cancer, and that is really the Li-Fraumeni that we are listening here for so many talks already. Briefly, for you, Lexis, this is how a cancer cell developed somewhere in our body. It's different. It has some mistakes in the genetic material, and most importantly, it doesn't know how to talk with the neighbors. It's proliferating and living as if he's only living in the world for himself. But that is not good, because when that happens, they don't control anymore their growth, and actually a tumor is something that looks very much like this, where you have a bulb or a globulus of cells which are proliferating, and what you described yesterday, a little pinhead, a little touch, all this is when the cells do not obey, the cells behave differently, and they most of the time generate a certain global lump, a certain tumor. So cancer can be seen as a cell which accumulates mistakes, and these mistakes are somehow interfering with normal control. But you would think nature must be probably perfect, because most of the time, even Li-Fraumeni people, they don't have that often, or not all the cells are sick, and there are probably mechanisms in nature which know how to control the development of cancer. And maybe we ought to look a little bit closer and understand how that is functioning in the body, and maybe try to mimic that to solve cancer at large. So basic researchers like me are really focusing on understanding how is it that a normal cell is converted to a cancer cell. So basically, you see, these are chromosomes. You heard so much about it. When you are a normal cell, you have these wonderful pairs, 23 pairs of chromosomes. When you are cancer, you see immediately chaos. In that case, there are two important things. We have this chaos in our cell, and in addition, we are losing the machinery, the natural machinery, which is constraining or inhibiting cancer development. And how is that being done? This is our DNA. We are exposed all the time to all kinds of stresses which damage our DNA. But that essentially doesn't call for cancer development, because our body is very smart. It developed all kinds of guardians, and these guardians are believed to be tumor suppressor genes. So there's a whole family of genes that take care that our genome will stay with great fidelity. So here you, for example, you see that is one such a guardian who's climbing on the DNA and looking for damaged DNA. These are the natural or our genomic guardians of the genome. The guardian of the genome can make two important decisions, like every smart guy. They go check for the DNA and assess if there's a lot of damage or if there's a little of damage. If there's a little of damage, they will consider, let's fix the damage. So they signal to a whole machinery of genes that will fix the damage and put on guard and fix the cell to become normal. But sometimes there's too much damage, like people go to the seashore, too much damage, and they have to do a different task. So let's see what they are doing. They can repair the DNA, and there's a whole molecular system that is called DNA repair machinery. And alternatively, when there's too much damage, they cause apoptosis, which is a word for killing, but a more sophisticated one, and this is how they get rid of the problem. So I'll just show you. Fixing is a machinery of taking care of the damaged DNA, and apoptosis is the Greek word for wilting. It looks normal, right? It's not a bad word, it's a nice word. You are fixing, you're taking care, you are removing the damaged cells, and you go on with life. So I'm standing here and you ask me, well, if this is the problem and everything is solved, how come we still have cancer? We still have cancer because our hero, the p53, which is the most important guardian of the genome, and this is a friend of mine, an artist, who made this picture for p53 after I told him the story. That guardian is like any other gene, undergoing a mutation. And if a policeman is undergoing a mutation, he becomes a corrupt policeman, cannot do his function anymore. So this is what is really happening. The important tumor suppressor, which has to do all these functions, that guardian all of a sudden cannot do his function, and not only that, not doing his function, the other way around is becoming, even helping the corruption, the performing or in turning into a cancer cell. So, talking about children, when I told this story to my granddaughter in Bal, I told her in Bal, so what do you say about my story? She said, there are two important friends, but not such great friends. The one is the good one, and that is the guardian of the genome. That is the wild-type p53. And later, you will see that basic researchers, like the next speakers, are very hard to understand how the wild-type p53 is working. And the second is the devil, and that is the mutant p53, and many people are trying to think of ways, how can you prevent the work of that devil? So that is, in a nutshell, the whole story of the mutant p53, our major enemy, and that is really occurring either in tumors in general people or in tumors, in germline tumors, which is Li-Fraumeni derived patients. So very quickly, I will just change a little bit the level, and David, please start counting my time now. So, we have somatic, a friend or not? We have here somatic mutations in tumors, and these somatic mutations are seen like very lazy guardians lying there in the tumor and doing nothing for the benefit of the tumor, the other way around. This picture I took, I think, in '81. And when a friend of mine came to ask me, and this was also raised yesterday here, what can we do with this? I said at the time, I don't think nothing, just watch and look. I think so many years later, we can do something. If you see the mutant p53, we already have some sword to find this enemy. So here you see that there are the germline mutations, and these are the mutations that are being transmitted in our genome. And actually, what stimulated my attention to this whole story was the 10 years after we discovered p53, and we were a very small group of maybe 20 people who thought p53 is important, and was very confused because we didn't know what is good and what is bad. We sorted out these questions, and finally, 10 years later, the story turned out from nothing to an important story, and that came really after the paper of David, who showed that this is really a genetic transmitted disease, and not only that, it is transmitted in human. I have to tell you that that year, we usually had maybe 10 or 20 publications per year. In 1991, we had a jump to 300 papers in the field, which told you how attractive the field became. And since then, everyone is interested in p53. So, the question that we ask is, what is really inducing the onset of the bad p53 in the Li-Fraumeni patients? We know that Li-Fraumeni patients are basically heterozygous, which means, and most of the time, they have one good allele and one allele which is mutated. That is the mutant allele, this one. And we have mapped p53, we know it's in chromosome 17, we have developed excellent tools, and we know to map the genome of p53 and today to find out the exact mutation. And you heard there's such variety and so much to work before we decide what to do. But basically, we are after the mechanism of understanding how the loss of heterozygosity is working. I would like to mention that what is happening under normal circumstances, a person who carries the mutation carries a good allele and a bad allele. This is the normal allele, and the one with the star is the mutation. And you can see that with time, there is a way that you can either lose the normal allele and duplicate the bad one, or somehow get rid of the normal region of the p53. But in any case, all of a sudden, you carry 100% genome that is coding now for mutant p53. So as long as a person is heterozygous, has a good one and a bad one, he is healthy. But the moment the good one is lost and the bad one is now turning into a duplicate, the disease is really coming out. And actually, this process of loss of heterozygosity is common and is found in all types of tumors. The sporadic tumors, which is just other p53 tumors, and in the Li-Fraumeni families. So we really wanted to understand how is that Li-Fraumeni, the loss of heterozygosity, connected to the outcome of tumor. So you probably heard that there were discussions and questions, and Pierre mentioned this morning the issue of how is that thing starting, and the concept of stem cells all of a sudden surfed in the air. And we become very much interested in asking the question, how are stem cells connected to the loss of heterozygosity, and all this, how is it connected to the onset of cancer in Li-Fraumeni patients? In other words, this is a theory that was accepted, that we have stem cells. Stem cells is something very good that takes care of the renewal of our body, in embryonic life, development, in adult life, renewal of all our systems. But then, if the stem cells, I'm sorry this is not working, if the stem cell is somehow getting a mutation, and especially getting a mutation of mutant p53, then this mutant p53 is turning a normal stem cell into a cancer stem cell, and we all believe that this is the origin of cancer. So we and others, the basic researcher, are now zooming on these stem cells and trying to understand how these stem cells are somehow associated with the loss of heterozygosity and the outcome of cancers in those patients. So our research goal pertaining to that subject is try to find a connection between stem cells, cancer homogeneity, and loss of heterozygosity. So to study stem cells, you can really approach it by a number of different systems. And let me just tell you briefly that in the mouse, you can work on the embryonic stem cells, eggs which are fertilized, and they contain the stem cells that are origin of life. This is where we develop and differentiate it to a whole body. And these are really the embryonic stem cells, which are mentioned here. The red one is an example of the embryonic stem cells. Excuse me, can you control that a little bit better? No. Here. So you have here the adults, you have the embryonic stem cells, we'll do it without it, embryonic stem cells, we have adult stem cells, and we have all kinds of technologies which develop stem cells. So we had several models that can look into stem cells. Stem cells mostly from embryonic life, and stem cells from the adult life. And we were asking the question, how is loss of heterozygosity connected to these stem cells? And is it really when loss of heterozygosity occurs, is that leading really to the cancer onset? So this is the model, and this is just to tell you that there are adult stem cells, and we are working on stem cells that are derived from the bone marrow, and you can work also on embryonic stem cells, and all these are just models that we pursued. So let's for a moment look on adult stem cells. Adult stem cells are derived from our bone marrow. We can generate them in the dish, and we can isolate them, and we can look what is happening when they have a good p53, when they have a bad p53, and when they have a situation of heterozygous, one good and one bad. Remember, when you have one good and one bad, the situation is not so bad. Tumors are not developed. So here is a way that we isolated these stem cells and got a number of stem cells of different natures. We developed a number of methods to follow how loss of heterozygosity is occurring. And maybe I will just show you very briefly here at the black box. When there is a Wildtype p53, there is a top band. And when there is a Wildtype p53 is the first one, that is the lower band. When there's a top band, then there is a mutant p53. And when there is two, it means one allele is good, one allele is bad. So we can follow and see if our cells express a good p53, express a good and a bad, or just a bad one. So we found, interestingly, when you take adult stem cells that are derived from bone marrow, and they are containing a good and a bad allele, and you culture them in the dish. With time, they are losing the wild-type allele, and they are left with the mutant allele. And we could show that when you have a mutant allele, these are mice which are born and they are modeling the Li-Fraumeni model. So when we take from them the bone marrow, we generate stem cells that contain a good allele and a bad allele, and we ask what's happening then. And we found that they lose the good allele, left with the bad allele, that is loss of heterozygosity, and that is probably modeling the development of tumor in a Li-Fraumeni patient. And indeed, when you take these, this is just to show you that they are undergoing 100% loss of heterozygosity, but now when you are taking these cells and inject them into mice, the stem cells which contain just the good allele are not making tumors. Stem cells which have a good and a bad allele are not making tumors. But stem cells which have lost the good allele, they are making tumors. So we found a good model that we can follow and understand what is the connection between loss of heterozygosity and tumor development. We are now pursuing this direction in very sophisticated method or mathematical method to understand what are the genes that are being altered in that program. And why are we doing it? Because we think that if we will understand how a cell is undergoing LOA and what are the genes that are involved in that process, we can prevent ways to block that process. If we will block that process, we will not let the cells undergo LOH and maybe stop the onset of stem cells that are undergoing LOH and maybe prevent tumor development in Li-Fraumeni patients. So that was our philosophy, and this is what we thought of doing, and actually we are presently doing that. But then, we thought that is one model. Let's go and look in another model of stem cells. And at that time, there was a Nobel Prize laureate, Yamanaka, who developed a system that you can take skin cells and make out of them stem cells, and that is, again, a very nice system to follow the development of cancer. So again, we developed that system and tried to look what is happening when these test systems have a wild-type p53, what is happening when they have a mutant p53, and what is happening when they are coming from mice, which are like Li-Fraumeni, that have one good allele and one bad allele. So I will just tell you briefly, because these slides can be a little bit complicated, what we found is the next. When we use this model of stem cell, which is a little bit different from the other one, we found that if you take a good IPS, which express wild-type p53, a good p53, they don't give you any tumor. The other way around, they develop into mice, healthy mice. If you take an IPS which contains mutant p53, they make tumors. But interestingly, when they are like the Li-Fraumeni situation, one allele good and one allele bad, there is a situation when they keep the two alleles. They make good mice and they develop like normal stem cells, but if they lose the mutant, they lose the good allele, they develop into tumor. So we had another sort of indication that in stem cells, when you have a wild-type p53, you are a good stem cell. If you have a bad wild-type p53, you are a cancer-making stem cell. But when in the middle, before I told you they look totally normal, but in this case, there is a certain incidence of such who are good and such who are bad. So very briefly, I will go through this data and show you that if you take these IPS, which are really expressing wild-type p53, the cells are normal to an extent that when you inject them into a mother, they will even develop a normal mouse. These are IPS. These are these very special stem cells, and they make mice which are normal. If now you take IPS which express the extreme, a mutant p53, you will see that they will develop very aggressive tumors. So our hypothesis was again, or confirmed our hypothesis, that indeed p53 is very important to control a normal life for stem cells. When it's not controlling that, it causes cancer. But what is happening now when you have heterozygous IPS, which is IPS or stem cells that are like the one that are coming from the Li-Fraumeni patient? In this case, we found the heterozygous one from a first sight looked normal. And when we made a more precise analysis of that, we found that there's certain incidents of them undergo LOH, and most of them do not undergo LOH. To tell you that stem cells have something that prevents an onset of LOH. So just to tell you that there were about 23% that did undergo LOH, and the rest were stable and did not go under LOH. So this is a very simple slide which shows you that indeed this is the profile of undergoing or not undergoing LOH, but what is really important is that the ones that undergo LOH are inducing tumors, and the ones which are staying heterozygous are not inducing tumors. So this is the one that is undergoing or not undergoing LOH, and the conclusion is that even when you have a heterozygous situation, somehow the good allele is dominating over the bad allele, and this is why they don't form tumor. So most of our cells, most of the cells in our adult life or an adult body of a Li-Fraumeni patient contain the two alleles, and this is why they don't make tumors. In order to make tumors, this has to occur probably in the population of stem cells which are in our body, and there has to be a process of losing the wild-type p53. So this is a very interesting situation, and maybe explains why in adult life we develop with time a tumor. The tumor is probably derived from stem cells, adult stem cells, and it's a product of losing the good allele and remaining with the bad allele. So that was the conclusion from that part of the work. And you see here, when we looked on the adult stem cells, it almost explained to us a situation. Why do they develop cancers in adult life? But at that point, we said, maybe we should go one step back. Because after all, you saw that when you have embryonic stem cells, doesn't matter what is their p53 status. If they are wild type, good ones, bad ones, or heterozygous, in all cases, they will produce normal mice. So we thought maybe that is the correct turf to understand how stem cells cope with this not losing maybe loss of heterozygosity, or losing or fighting with the mutant p53, which is a very interesting situation. Maybe I'm a little bit complicated, but I will just tell you that from that point, we embarked on the stem cells, and we found an interesting thing here, and that is, because I have only 3 minutes, that embryonic stem cells do not undergo LOH, which is important, but more important, we found that even when they are totally built of mutant p53, which is we made in the laboratory now. If you have a good stem cell and it's good, fine, and it doesn't undergo LOH, is also fine. But what happens when you have a stem cell that has a mutant p53? It doesn't have a good allele at all. We found that in these stem cells, there is a certain system that is shutting down the mutant p53. And the question was, how come? So I will just tell you briefly how it happens. It happens so that the body will do everything in the embryonic life to shut off the expression of mutant p53. And in this case, we looked into the proteins of the cell, and we found that embryonic stem cells have a very interesting machinery of proteins which are complexing with the mutant p53 and preventing it of becoming a mutant p53. Amazing. Theoretically, you have a sequence that codes for a mutant p53, but practically, even if the genome is 100% coding for a bad p53, it cannot express itself to become a mutant p53 molecule. And that was really the basis of our work to try to understand what is the mechanism that is really preventing a mutant p53 from exhibiting a nature of a mutant p53. So we developed here, and this is why I really want to take 2 minutes, a new methodology of how to convert the mutant p53 into a wild-type p53. So the idea was, in nature, in embryos, there is a mechanism of proteins that know how to take a mutant p53 and convert it into a wild-type p53. So this is a wild-type p53, well folded. And when the mutant p53 is like that, it's not folded and it's not doing its function. So what we were looking for is for proteins that will bind to the mutant p53 and refold it into a wild-type p53. And that is really the strategy that we developed for the last 3 years. And the strategy is very simple. It is based on taking mutant, the M, and converting it to the wild type, the other way around. And that is by a method of interacting it with small peptides. There has to be a certain handle that will attack that to become that. And there is in the literature ways to show you that sometimes like little pins can touch a protein and it will refold itself back to a normal configuration. So actually what we did is we used a library of small peptides and screened for such peptides. And I will make a very long story short. 3 years worth of screening, having excellent reagent developed in the field, we came up with a group of protein, which we call them our lead team of proteins. And these proteins, we could show that these small peptides, we can show that when they interact with the mutant p53, they really fold it back to a good p53. So this is an excellent tool. A lot of work into that, a lot of basic research, and I would just want to show you that this is how we envisage the p53 structure. We looked on it, we crystallized it, and this is the upper band here, the green thing. I'm sorry. No. Can you just take it back? Because it doesn't go back. Back. Another back. This is really the molecule. And what are we doing with this molecule now? Obviously, we could show that it works in the culture. We can take mutant p53, sprinkle on that the small peptides, and convert it into a wild-type p53 and a good p53 by many different assays. But what I really want to show you is this simple experiment where you take an animal that had already developed a tumor, inject into the tumor that peptide, and the tumor disappears. You don't have to be a molecular biologist or even not an animal biologist to see that with your eyes. And here's another example of a group that we treat with a control and a group which we treated with our desired protein, where we change now the mutant p53 into a good p53, and we believe that we cause rejection of tumor. So our future is really to develop a way to treat cancer patients with peptide. But what is the punchline? What I really want to tell you is that I believe that with these small peptides, which are very naive materials, can be regarded as food additives. What we would like to suggest this forum to consider the possibility that we can use these small peptides as a food additive, as a preventive way to be given to Li-Fraumeni potential patients, so to prevent the expression of mutant p53 in such patients. And this is where I leave you with this idea that me and my friend Moshe Oren and other people at the Weizmann Institute are not trying to launch that project and hope that maybe through our friend David we can come across the audience of Li-Fraumeni patient and maybe think about this possibility. And actually, as was shown yesterday by Amanda, I leave this open page for you to come up with ideas and thoughts. How can you help us really promote such a project? We worked on it together, me and my partner, a work partner, 60 years of basic research. The last couple of years, we really turned into be translational people, which is to do something for humankind. And now we are in this open page as if step one, where we want to push the whole idea of trying to use it as a preventative drug for Li-Fraumeni patients. And obviously, I would like to thank all the people who contributed, my students, my postdocs, and if you know here, David Malkin from Toronto. There is Peter Lichter from Heidelberg, who's helping us a lot with the sequencing data. Tammy Geiger, who is the woman who did the wonderful work on isolating these proteins, which inspired us to do this method. And obviously, Dov Tzipori, who was a neighbor in Toronto and developed the idea of stem cells, and all other students. And I'm sorry I took a little bit over time, because I'm always so excited to tell my story. I'm sorry. Nobody wants to know if we can give out the protein, the peptides. Yes. We are working on it.
Where do you sign up, right?
What?
Patients. Where, I mean, if you're wanting to develop a study, right?
We want to apply it to human.
Yes, and so as patients, I know, sign me up, right? So how will we follow your work so that when you are ready to enroll people, that we can do that?
If you give me a consent, I will update David the moment we are ready.
Okay.
He will be the first one to know.
I think the idea here, and I think this will come through the afternoon, is we're going to hear, as these novel peptides and things come into place, your question's well positioned. I mean, I think the idea of having it ready for a patient today, no, but it's to have the system ready and primed so that when it does become available for human, quotes, consumption, we're not then waiting for the FDA and for all the other regulations to be another 3 or 4 years before it's actually ready. So I think that's where there is an opportunity for the various advocacy groups that are represented here to really start working with us, to work with the FDA and the regulatory parties in other countries to have the system ready to go, because I have a feeling there will be a lot of these, right?
There are a lot of these.
I was going to ask you, how do you give... what's the preparation that the peptide is for the mice? Is it by oral or?
No. You would be surprised that we buy the peptides from all over the world, even from China, and we apply them in many different ways, and we are now working on modifying them somehow so you can take them peri-host, you can take them IV, and now what we are doing with the mice, we are either injecting intratumoral or subcutaneous. And obviously, in the mouse, we are already exhausting the situation. And actually, we are already using human ovarian cancer, which has 100% penetrance of mutant P53, as a model in mouse. Primary tumors, xenografts, and all kinds of therapies.
Thank you. Okay, we'll move on to...